Hello, welcome to Vega Achiever Educational Limited. Presenting, we are tutor, trusted from brain friendly and high impact learning. Still on transport and cell biology. In our previous class, we discussed about transport and cell biology. We talked about the types of transport and cell, which include diffusion, osmosis, and active transport. We've discussed about diffusion, so now let's continue from the other part, which is osmosis. Now, what is osmosis? What comes to your mind when you hear the word osmosis? And you should know that osmosis is the movement of water molecule from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. Or we can also see that osmosis is the movement of water molecule from higher water concentration, which is in dilute solution, to a lower water concentration, which is concentrated solution, across a partially permeable membrane. And it is passive. Being passive means that it does not require energy. Now, let's talk about the terms used in osmosis. Now, there's something known as tonicity, and these are types of solution in osmosis. Now, you have the hypotonic solution, the isotonic solution, and the hypertonic solution. Now, what is hypotonic solution? Hypotonic solution is a lower solute concentration, higher water concentration, which simply means the level of solutes is low. The solute can either be salt or sugar, is low, and the higher water concentration is very high, which simply means it contains high amount of water when compared to the solute present in it. That is what hypotonic solution means. I haven't talked about that. Now let's go to isotonic solution. Now, what is an isotonic solution? Same solute concentration as inside the cell. Now, what does this mean? It simply means that the numbers of solutes or water present within a cell is in equilibrium with the cell environment, which simply means none of them is higher than each other. They are in equilibrium state. That is what isotonic solution means. Then as for hypertonic solution, it means a higher solute concentration and a less water concentration, which simply means the level or the amount of solute is higher than that of water. When you compare these to hypotonic solution, you discover that in hypo, solute was low and water was high, while in hyper, solute is high and water concentration is low. So have that in mind. Now, let's talk about osmosis in animal cell. After discussing that of animal cell, we also discuss that of plant cell. Now, osmosis in animal cell involves placing an animal cell in solutions. The solution may be hypotonic or isotonic or even hypertonic. Now, what happens when an animal cell is placed in an hypotonic solution? Now, remember, in hypotonic solution, the solute is low and the level of water is high. Meaning, in this case, water is going to move from a region of higher concentration towards lower concentration. Since the animal cell is placed in an hypotonic solution, it simply means that the solution have low amount of solutes. Hence, water will leave the solution and enters into the cell. And when water enters into the cell will swell and also burst. That is called lysis. Now, the cell bursts. That is the bursting of the red blood cell is known as hemolysis. Hemolysis is the bursting of the red blood cell, because an animal cell, when placed in an hypotonic solution, water enters. That's number one. Number two, the cell swells, which is number two target, and number three, it bursts. Now, I haven't talked about hypotonic solution. Now let's talk about isotonic. Now, in isotonic, no net movement of water, cell remain normal. It simply means they are in equilibrium. Now, what now happens when an animal cell is placed in an hypertonic solution? Now, this is what you need to know. Now, since it is placed in an hypertonic solution, it simply means that the level of water is low in the solution and the solute is high, which means water will leave the cell and enters into the solution. Now, when water leaves the red blood cell and enters into the solution, the cell shrinks. That is what you call crenation. Now here is the diagram that shows isotonic, hypertonic, and hypotonic solution. Now let's start with the hypotonic solution, which is the first diagram at our left. Now, in the hypotonic solution, what do you notice? Water enters into the cell, which simply means the cell swells, and after it is swollen, then what happens next? It bursts. Now, mind you should take note of this. The red blood cell is going to burst when placed in an hypotonic solution because they do not have a cell membrane which can help them withstand the pressure of water inside. Now, going to isotonic solution, you can see that they are in equilibrium, or no net movement of water. And you can see the blue arrow there shows that water is entering into the cell in hypotonic solution, and in hypertonic solution you can see water leaving the cell, while in isotonic solution, which is the middle one, you can see both water enter and leaves the cell at the same time because they are in equilibrium. Whatever is lost is being gained. That's it. But in hypertonic solution, if you notice, water leaves the cell in hypertonic solution and the cell shrinks. Yes, and the shrinking of the red blood cell is called crenation. Now let's talk about osmosis in plants. I haven't talked about that of animal. Now let's talk about osmosis in plant cell. Osmosis in plant cell. When a plant cell is placed in an hypotonic solution, water enters the cell. The plant cell, it is swollen, then becomes turgid, thin. Unlike animal cell, won't become swollen, it bursts. Plant cell won't burst because they have cell wall, which help to keep the structure of the cell intact. Now, talking about isotonic solution, as usual, the net movement becomes flaccid, meaning soft, in equilibrium state. Then, when a plant cell is placed in an hypotonic solution, what happens? Water leaves the cell. And when water leaves the cell becomes plasmolized. Now, why does this cell become plasmolized, or what is plasmolysis? It is the process in which water leaves a plant cell and the cytoplasm is pulled away from the cell wall, which means pulling away the cytoplasm from the cell wall means plasmolized in plants, which means the plant cell have become plasmolized when placed in an hypertonic solution. Now, this diagram shows the hypertonic, hypotonic, and isotonic solution of a plant cell. Now, if you can see, the cell wall is there, no net movement of water into or out of the cell, meaning when the cell is not immersed in an isotonic solution, what happens? It is in equilibrium states. Now, looking at our left-hand side, when water leaves the cell shrinks and become plasmolized. If you notice, the cytoplasm of the cell is being pulled away from the cell wall, forming a structure that looks like a star shape in that place, meaning that cell has become plasmolized when placed in an hypertonic solution. If you check at the left-hand side, and on the right-hand side of the screen, if you check when placed in an hypotonic solution, it becomes turgid, which simply means no effects, like it becomes tightened. Yeah, that's it, and it won't burst because of the presence of cell wall. Now let's talk. Okay. Here is the summary of what we've learned so far. Now you can actually take your time to look at it. You can see osmosis right there, tonicity, hypotonic solution, isotonic, hypertonic rather, and different effects on plants versus animal too. Now here's also some key difference between animal cells and plant cells. If you notice, animal cells lack a cell wall, can burst or shrink easily. Plant cells have a cell wall, prevent bursting, but can plasmolize, meaning pull away from the cell wall. The routine plant provides support for the stem and all that, and you can see that of animal didn't have cell wall though. Now let's answer some GCSE questions. Now the first question on our list is this. It said, use the words from the box to name the structure labeled A and B. Number one, it said cell membrane, chloroplast, cytoplasm, and nucleus. Anyways, A represents the nucleus and B represents the cell membrane. And question 2 then says the cell in the diagram is an animal cell. How can you tell it is an animal cell and not a plant cell? Give 2 reasons. Number 1 reason is that is an animal cell because it does not have a cell wall. If you notice, you already have a cell membrane there. And 2, it does not have a large vacuole. Yeah, it does not have vacuole, which is present in plant cell. So animal cell don't have that. Now the next question on our list is, a student puts red blood cell into water, suggest what will happen. Yeah, this is very easy. And in this case, water will definitely move into the cell. Since it's a red blood cell, water will move into the cell. And what happens when water moves into the cell? The cell is going to definitely swell up or swells. And after it swells, what happens next? It bursts, which is lysis, or called as hemolysis, the bursting of the water of the red blood cell. So So that is it. For more information on this topic, contact us as Vega Achiever Educational Limited. Thank you.